on evolution. We are delighted to have Dr. John Hawks, Assistant Professor of Anthropology at the University of Wisconsin-Madison. And Dr. Hawks. There you are.
Hi, thanks everybody. And I want to sincerely thank the organizers for this invitation. It's a really exciting place to talk about what I do. And the last talk, especially the last part of it, was a great introduction to my work because we're using these data that come from microarrays ultimately to talk about the evolution, in my case, of humans, but in a broader sense, the evolution of everything. Understanding genetically how things are connected to genes. I'm working at this from sort of the opposite end. I don't sequence things in my lab. I don't run microarrays. I use these data to try to find out what's happened in human history. So I'm going to give you some case studies about what kinds of things have been changing in human evolution. I'll try to give you a broad picture of the rate of recent evolution. That's been really the focus of my work the last couple of years. And also try to touch on some of the things that have really changed about the way that we look at human evolution from the anthropological perspective, given what we know today about human genetics. All right, so those of you who saw the title of my talk, Can Genetics Explain Historical Events? You might have thought of maybe the most obvious case of this. Who is this? Yeah, Rasputin, right? Rasputin's effect on history is a genetic effect, and most of you know the story. There were these people, the star-crossed lovers, Alex and Nikki. Alex, unbeknownst to anybody, was a carrier of a rare genetic disorder, which just a couple of weeks ago was characterized by genetic sampling of the bones of these people and their offspring as a factor IX hemophilia. The son, Alexei, who would have been Tsar, where's the son? There he is, right in front. The son who would have been Tsar inherited this. It's X-linked so that you only have one copy of it. If you're a female, you're a carrier. You don't have the hemophilia. But if you have one copy of it as a male, you only have one X chromosome. So it's not masked. So people who carry one copy of it have the hemophilia. Of course, the family consulted with Rasputin, who had sort of this odd effect on people. There are all these pictures of these Russian tea parties with all of these prim and proper women, and then there's one person who doesn't fit. I show this in my classes. There are 4 or 5 of these pictures like this. But he had this spell over Russian women, evidently, and was a faith healer. Thought, I could deal with the Tsarevich, and of course failed. This is one way that genetics have affected history. It's not the way that I'm especially focusing on, but I think it's a great case because it's in the news. Queen Victoria was the originator of this allele. It's a very rare kind of hemophilia. And they've traced it through the genealogy of her family. Her scheme, of course, was to get all of the royal families of Europe to be her descendants so that peace would prevail. And she succeeded in this substantially. You can see the names of all these royal families of England up here, including Kaiser Wilhelm and her own son, the king, and the Tsar of Russia. They're all her descendants, right? Peace did not happen, but hemophilia did. And it also affected the royal family of Spain, where 2 of the sons, we believe, were affected by it, and 2 of them died in car crashes, leaving the Spanish succession in doubt and possibly fueling the Spanish Civil War. So genetics has a direct effect on history. That's the way that individuals affect history. I want to talk about the way that broad trends affect history. And for this, we have to go back in time a little farther and think about the things that we have that are good genetically. Lots of times when I'm talking about genetics, it's always disease this, disease that. Like the genes are there to make you sick. They're not there for that. They do good things for us too. And one of the things they do is to make us into super mutants. Here's Superman. He's from planet Krypton, but his milk drinking ability is distinctively European. This is something that's widespread in West Eurasian populations. It's also widespread in sub-Saharan Africa. It is in the Arabian Peninsula, and we now understand from genetics that there are 5 different versions of this gene that have been selected in the last 10,000 years. Within the last 10,000 years, milk drinking, the ability to do this as adults as opposed to as kids, so all mammals drink milk as kids, but to retain this lactose digestion possibility, it's something that's entirely new in our species. It's not shared by most people. As far as I know, Superman does not have any immunity to malaria, but many people in sub-Saharan Africa have a partial immunity. Here's the Aedes mosquito that transmits it. Here's what malaria parasites do to your blood cells. It's bad. It's the leading disease, infectious disease, cause of death in the world today. It's bad. It's so bad that it has prompted genetic responses to it. The most well-known of those is the sickle cell response. It's well-known because it's in all of our textbooks, because it's weird, right? It's bad for you if you have 2 copies of it. It's good for you if you have 1 copy of it if you live somewhere with malaria. If you have 1 copy of it and you live at high altitude, it's not necessarily so good. So it's a well-known case because of this selective balance. But in fact, there are probably about 24, or maybe more, genetic responses to malaria in sub-Saharan Africa. And there are other genetic responses in other parts of the world that have historically been high malaria regions. This is a cause of mortality to which humans have developed defenses. Falciparum malaria, the worst kind, the kind that kills most people today, it's only 5,000 years old. So we have genetic responses to this disease that have appeared within the last few thousand years. This is my daughter. She's pretty blue eyes. Blue eyes are caused by a change in the regulation of a gene called OCA2. OCA2, oculocutaneous albinism type 2. It is a known gene because it makes some people in sub-Saharan Africa and in the southwest United States albino. But it's not a disease that makes you sick. It's a disease that regulates pigmentation. And there are a number of genes that are part of the pathway to do this. OCA2, the regulatory mutation that looks like it makes blue eyes, we estimate to be about 10,000 years old. 10,000 years ago, no one on Earth had blue eyes. Okay, you guys got Rasputin. Anybody know who this is? I don't think he's grateful for being dead. This is Robert the Bruce. This is the skull of Robert the Bruce. In anthropology, I've got to tell you that I interact with people who, and I'm one of these people who feels skulls to try to figure out what's going on with them. And to make the transition from genetics to traditional anthropology and back, this is quite a challenge. I'm going to try to put these things together for you to the extent that I can, because these are 2 different ways now that we have of looking at the past. The traditional way, feeling the skulls, leads to a lot of conclusions about what's happening in the recent past in human populations. So for example, this is a chart of femur length, the length of your thigh bone, versus time, where here, in opposition to almost all of my slides, here I've got 0 at the left side and 10,000 years into the past on the right side. You'll see that I make these negative most of the time, so that the past starts at the left and comes to the present at the right. What do we got here? We've got essentially a chart that's showing no change to the extent that we can see. These are the regressions. This is a chart that shows brain size in the same population. Now, this is the population of southern Africa. This is this is the ancestors of today's San populations. What's happened to brain size is that it has shrunk, and this regression is males and this regression is females. It's shrunk over the last few thousand years. Brains are shrinking. Southern Africa is in no way unique. I put it first because I wanted to make that point of body size. Brains are shrinking, the body is not changing in size. And also to make the point that this is worldwide. Here's Europe. Europe, this is endocranial volume means in males on the top and females next. The only disjoint here in this distribution is I don't have a sample of Roman era females to go in and spot. What happened? Early Upper Paleolithic people, people who lived 25,000, 15,000 years ago, had bigger brains than Neanderthals. A lot of you who know anthropology have heard Neanderthals had bigger brains than us. It's true. Neanderthals did have bigger brains than us, but they didn't have bigger brains than the people who followed them in Europe. Those people's brains were expanding until about 10,000 years ago when they started to shrink. The shrinking that's happened since 10,000 years ago, they've gone down in males and females by about 150 cubic centimeters. If we went down by another 150, we would be Homo erectus size. This is a big change. The standard deviation in endocranial volume, this measure today, the standard deviation in this is about 87 cubic centimeters. So we've changed by almost 2 standard deviations. Other stuff has changed too. Here's the thickness of the skull. This is a simple, you put the caliper on the inside and the outside, you measure the thickness. Thickness has reduced. Our skulls have gotten more gracile. And if you study 10,000 or even Bronze Age era European crania, this is Europe, you'll notice that they're much more robust looking. Robert the Bruce was no exception in his population. That was a skull that had some rusticity to it. Coincident with this change in brain size has been a change in the ratio of the length to the breadth. This ratio is one that students don't study very much anymore, but that used to be what people really knew about humans, the cranial index. You would go around with a caliper and you measure people and you tell them about themselves. Oh, there's a famous part of On the Town where the museum lady is, oh, you're dolichocraniocephalic. That's exactly what this is. Cranial index. Bigger cranial indices mean that the head's broader compared to its length. Smaller cranial indices mean that it's longer compared to its breadth. So the head changes in shape. Now, why do I show you this old-fashioned measure of human variation? I'm showing you this to show you that it's changed a lot. The heads have gotten broader. Why have this changed? It's probably changed this way, I think, although I can't prove it, because the heads have gotten smaller. It's gotten smaller, and it's affected the length disproportionately. But the interesting thing about it is that in the olden days, 150, 100 years ago, this was the logic of human race differences. Why did people care about the long heads? Because it was the invasion of the long heads that explained the superiority of the Nordic races. We don't talk about the Nordic races anymore. We don't talk about the invasion of the long heads anymore. This didn't happen. It's completely fictitious. But the measurements are still there. This is something that has changed in recent human populations. This change was recognized by Franz Weidenreich, who was known as a great anti-racist in his day, but also was the person who studied Peking man. That's why he was famous. And this is a chart that shows Sweden and Denmark head indices in 2 samples, Neolithic, that's early farmers, and modern day. And what you've got is a change in the average going from or higher, and in the next slide of Swiss, it's more evident. These are Roman era, 5th to 8th centuries versus 19th century. The cranial index changed. This is evolutionary change in the skull. Now, when we talk about genetics, we can look at genes that have changed in frequency in recent times and infer something about their history. When did this mutation happen? How fast has it spread? That's something that tells us a tremendous amount more information, in a sense, than this kind of information. And yet, these people, they were buried in the ground at that time. So they tell us a kind of information that genetics can't. The challenge that my lab works on is, how do you connect these things? Oh yes, endocranial volume also decreasing in China, and teeth are reducing in size. Recent evolution is real, and it's stuff we can see by looking at archaeological samples. Okay, a lot of you know what genes are. A few of you I know, this is a little outside your range. So I just want to give a couple words about how we study genetics today from my point of view. This is not the wet lab point of view. This is maybe more of an informatic point of view. So we've got Genes, how do we find them? What are they? How do we look at them? What visual picture should we have when we think of a gene? For a lot of us, it's this, chromosomes, and they're real. You look at them through an electron microscope. There they are. This is a genetic map. This is showing you the same things that you just saw, chromosomes, but here what you've got is a schematic that 1, 2, 3, 4. Many of you may not know that you can see that they're in order. The 1 is biggest and 22 is the smallest. It's because that's how they were numbered. Biggest one, number 1, 2, 3, and so on. That's why chromosome 1 is 1. They have these bands on them. These are banding patterns that were originally induced by staining them, but we still use them to identify the locations of things on chromosomes. If something is linked to the inheritance of one of these bands, we say that's the region where it is, something like 2q22.3. And here's a little more detailed genetic map. This is what you can get on genome browsers today. These are open access. You can find them on the internet. You can search for genes. I get all of my students to do this nowadays. Here's chromosome 2. We're looking at one little part of chromosome 2, the little red bar there. And this is the lactase gene, LCT, the gene that makes lactase. It's got reading frames, exons here, and big spaces in between them. This is also showing you, I want to point out, some stuff that we know about genetic samples of humans. For one thing, we've got the same sequence in chimpanzees and orangutans. This is just showing you that there are overlaps, that yes, these parts are covered if you wanted to compare them. And then we've got single nucleotide polymorphisms. Those are the SNPs that you heard about in the last talk. These are places where one copy of a gene has one allele and another copy of the gene has a different allele in human populations. So single nucleotide polymorphisms. Most of my inferences today are based on the SNPs. You can click on those and find out information about them too. This is really cool. And there's a lot of them. There's a lot of SNPs. The samples that I'm using are about 3.5 million SNPs, genome-wide. Okay, if we zoom out a little bit, you can see what's next to lactase. And here's lactase here, still in the middle, highlighted. And I wanted to point out this gene next to it, MCM6. Has nothing to do with milk, cell membrane something or other. And this arrow is pointing to an intron of the gene that has a mutation in it that makes Europeans drink milk. So there it is. That's the spot. It's a downstream mutation, but it regulates this gene. Okay. Now, one of the things that we care about in terms of understanding the evolution of these genes is, are they neutral or are they selected? Do they do something that's good that makes people reproduce more, that is, they increase fitness, or do they do something that evolution really care about and it could take it or leave it? I'm interested in the things that are selected because, as you'll see, they impact our understanding of the demographic forces in the population, how it's changed over time. And of course, we care about these things. They're cool. They're things that show us the opportunities for adaptation in our lineage in the past. But almost everything that we study in terms of these polymorphisms, almost everything is neutral. That's very convenient because it means that we can use the things that don't do anything to try to understand the things that do. So let me give you a schematic of how the evolutionary process works. We study changes due to new mutations by linkage. Linkage is due to the physical connection between polymorphisms in one place and polymorphisms in other places. What you discover is that things are inherited together because recombination breaks up chromosomes so that you inherit different parts of them from your mother and from your father. They reconnect. But every so often across a chromosome that's broken up and recombined so that you have a different new version that contains elements of your father's and elements of your mother's to pass on to your offspring. That's recombination. All right, so what happens if we introduce a new mutation to a sample of genes? Well, first thing that happens is it's here. If it survives, and the likelihood of it surviving is not real great, it may increase. If it increases, what happens? Well, notice what's happening is that this copy is increasing, and it carries with it the stuff that it's connected to. This is linkage. This process we call hitchhiking. Basically, you're starting out with one thing, and these other polymorphisms, the allele that happen to be on the same chromosome as this one, they get replicated along with it. So they're hitchhiking along toward higher frequency. When something gets up to this stage, and we have a big sample of people, we can notice this. You say, hey, look, everybody who carries this also has this, 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 and this. Whereas, if they don't carry that, well, look, some of them have this, and some of them have that. Some of them have this, and some of them have that. They're all mixed up. We can compare the mixing among the people who don't have it to the uniformity among the people who do have it, and that tells us something. What it tells us is that this thing didn't used to be very common. As this proceeds, as the evolution continues, what you'll notice is that every so often recombination happens, and what's out further away than this recombination event, now this is no longer exactly the same. Once it proceeds all the way to fixation, more recombination events happen. What you discover is that there's a region of the genome, it may be a relatively short or long region, depending on how long this process takes, that's uniform, where we don't find polymorphisms very much. And then, there's, as you go out farther away from it, variations increase. It's at this stage where we can really find things easily. And it's at this stage where we can use the process of recombination to try to figure out how old things are. When I say something is 10,000 years old, what does that mean? It means that I've discovered that there's an area of the genome where people who carry one haplotype, one allele, are depauperate in variation, and the length of that region, before recombination is shuffling it up, the length of that region is consistent with an age of 10,000 years. So here's an example of one of these. This is from my colleagues Bob Moises and Eric Wang. What have they done? They've compared people who carry one, the yellow copy of the central allele, with people who carry the other, the blue copy. This central location is the one that they're studying, but then they look at other SNPs further along the line to see whether they're identical or not. This is a measure of homozygosity over space, in other words. And what you discover is that people who carry the yellow one are identical. There's no variation over a long region, whereas people who carry the blue one are substantially mixed over this range. This is unusual, and it's an inference that this gene, reticulon, is recently selected. You get different patterns for different genes with respect to linkage over distance, because some of them, like G6PD, glucose 6-phosphate deficiency, this is the major malaria defense in sub-Saharan Africa. It's X-linked, and it's a little bit bad for you, but it's very recent. This has probably emerged within the last 2,000 or 3,000 years. Versus this one, DRD4, this is the D4 dopamine receptor. The 7R allele is an allele that is interesting because it's associated with ADHD in clinical subjects. So it gives you a little bit higher likelihood of having ADHD. It looks like it was selected, but we'd say a lot longer ago because it extends over less space. Our best guess for this is within the last 40,000 years as opposed to within the last 4,000 years. So this is giving us a gross picture of the ages of mutations that have been recently selected, and it's giving us some picture of what's going on in our evolution. Here's another way of looking at recent selection. This is the geographic distribution. A lot of times when you read about selection, it's about why populations are different from each other. If we assume something about the history of populations, how big the populations were, how long they've existed, then we can predict things about how different they ought to be. Human populations on average, if we compare alleles in Europeans, Asians, Africans, Native Americans, those alleles have about a 0.1 chance of being shared within a population versus being separated between populations. That 0.1 chance is what statistic we call FST. And if we see an FST that's really, really, really high, something like 0.8, so that's odd. This gene has this strange geographic pattern. It's really different between regions. Here's a gene that's like that. This is a SNP in a gene called SLC24A5. Here's the derived version of it is the orange here, and the ancestral version of it is the blue. And this is showing you a pie chart for each of many populations of the frequencies. You can see that the ancestral version is 100% in most of East Asia and in most of sub-Saharan Africa. The derived version is up close to 100% in Southern Europe and at high frequencies as far as Western India and Pakistan. What's going on with this? SLC24A5 is the single biggest gene influencing skin pigmentation in Europeans. This is a light skin gene. I say a light skin gene because there are a lot of them. There's as many as a dozen in Europeans, and there's a dozen in Asians, and they're different for the most part. This is one case. North Asians have light skin. They do not have light skin by the mechanism of SLC24A5. I wanted to put this one up here to show you that in order to identify selection this way, you sort of really have to have the mutation that causes it, or at least one that's very, very tightly linked to it. Because here's a SNP also in SLC24A5, but just a little bit downstream. And you can see that this SNP doesn't have the same extreme geographic differences. Does have a geographic difference, but they're not as extreme. It's the selected things that have this really odd difference in this case. All right, this gene I'm going to show you, this is a SNP in MC1R. MC1R is the redhead gene. But this allele doesn't make you redheaded. This is a functional mutation that probably, although the mechanism isn't known, probably makes lighter skin pigmentation in East Asians. I say probably because it's selected, it's recent, and it's very different between populations. Europeans who have red hair have different functional mutations, and there are 3 of them that are correlated with red hair in Europe. So here's one gene, different alleles in different places, all of them probably selected for pigmentation variation. This is interesting. Our species is evolving like crazy in pigmentation in different ways in different populations, presumably because of the same underlying selection pressures. And there's sort of a more, I don't know, this is so cool and yet so old school. How would you really want to know how things changed in the recent history? Well, the way that you really want to know is to dig up bodies and see what their alleles are. And of course, we're able to do that now to a substantial extent. So here's a chart this is a map of basically Europe. These green shaded areas are areas of early agriculture. This is from a paper that came out about a month ago, not from my lab, from Bramante and colleagues. And I wanted to show this to you because it's so interesting. What they've done is they've sampled the ancient mitochondrial haplotypes of people, and these bubbles show you where they sampled and the number of samples that they had. They sampled early farmers, people buried in early Neolithic contexts, and hunter-gatherers who'd lived there right before the early farmers, and modern people who live there now. What they discovered is that the early farmers don't look like the hunter-gatherers. Hunter-gatherers are mainly haplogroup U. Early farmers have a very low U and high frequency of N. Well, that's interesting. It means that agriculture, as it came into Europe, was bringing new people into Europe with it. It was not being adopted by the people who were there before. But the other cool thing is that these Neolithic people, with their high frequency of N, today, N is virtually absent in Europe. These Neolithic people aren't today's Europeans either. So you've had 2 population changes. Now, how does this happen? We've got processes that are competing with each other to explain this. A, migration. People picked up, they brought their farming with them, they put down, and then later on some other people picked up and came in, plunked down. And you've got selection. The farmers came in, maybe they were bringing new genes, maybe not, but some new gene worked in that context. And then later on, the new gene didn't work anymore, or a different one worked better. And so you have a genetic replacement as a consequence of fitness, as a consequence of reproduction. This is my job to try to sort these out. I won't say I've sorted this out yet, but I'll tell you the dynamics that underlie it. For example, when you have a new mutation, one of the things about it is that it, if it's selected, increases extraordinarily slowly for a long time. Our best estimate of the age of the lactase version in Europeans is about 8,000 years old. 5,000 years ago, these early Neolithic skulls, or skeletons, the ones that were genetically sampled, they don't have it. It's not found in them. So about 5,000 years ago, introduction of agriculture into Europe is not there. That's exactly what we expect. It takes a long time for this to get to any substantial frequency. And if you sample 25, 30, maybe 50 people from that time, you're not going to find it. Today, this is about 80% in parts of Northern Europe, which means that the number of lactase intolerant people is about the square of 20%, or about 4% in Northern Europe. That's about right. It means that 80% of today's Northern Europeans, 80% of the population of Germany, got this gene from one person who lived 8,000 years ago. What's the population of Germany? Something like 60 million, right? So you say 80% of the 120 million copies of lactase in Germany now came from this one person. But I'll tell you something else about this, and something that's very interesting, is that there's 40 million copies of this gene floating around in Germany now that are descended from the people who had the non-tolerant version. There were not 40 million of those genes 5,000 years ago. There probably weren't 10 million of those genes. So both versions of this have undergone a massive increase in population. One more than the other. One's gone from 0 up to 80%. The other one's gone from not very many to many. All right, so a kind of thing we can look at is if we look genome-wide and ask how many of these things are there that look like they've increased in frequency a lot lately, how many? It looks like about 3,000. This is a lot of them. Different populations. This chart is showing a blip for every one that we found that has this signature where it's a linkage disequilibrium signature. And a couple of things, these little lines, these are chromosomes, each block, and these little lines are showing different samples, Europeans, Chinese, Japanese, and Yoruba. Those are the major HapMap samples. And what you discover is that sometimes they're shared. You see a lot of them there shared. Sometimes they're not. Different ones in different populations. That's a clue of what's going on. The other thing is that the sheer number of them. When I started this work in 2006, nobody thought this was unusual. Well, 3,000 genes under selection? Well, who knows? How many should there be? The 3,000 genes under selection, let me tell you, there are 40,000 amino acid changes between humans and chimpanzees, and probably only about 12,000 of those are selected. So what we're saying is that a fourth of the potential differences between humans and chimps are underway now in the human population. That's like 3 million years of evolution packed into the last 20,000 years. That's crazy. So I got interested in this. What's going on? An obvious kind of answer for what's going on is to look at what's happened to the human population in recent times. And the number one thing that's happened to our population in a global sense is that we've increased in numbers. That increase was mainly Neolithic, post-agricultural, and later. So looking at this Neolithic time period, we might expect that there would be some really interesting stuff genetically. This is a time when people are changing their environment, after all. They're adopting new foods. They're living in cities for the first time. They're expanding to new places where people had never lived before. But also, there's just a lot more of them. But population increase didn't start then. It started earlier. We know that archaeologically. We do things like, this is sort of silly stuff, like we measure the size of tortoises. But the thing is that people eat tortoises. And the more tortoises that people eat, the less old the tortoises get. And so what you find is that when the population grows, you get a lot of little tortoises. And here you can see the tortoise size declining as we go forward in time. And people broaden their diet. So archaeologically, we know that populations were growing before agriculture. And of course, this is the major explanation for why people invent agriculture. Population pressure. They've got to find new foods. So if we look at a schematic of how the population's been growing, it's been growing gradually, at least, for a long time, certainly after the expansion of modern humans. The African population was always quite a lot larger than populations outside of Africa until the Neolithic, when things sort of reversed for a while, because agriculture is invented in primary centers, China, West Asia, ultimately the New World. Africa gets agriculture a little later, but then it does expand quite rapidly. I wasn't the first person to think of this, and how could I be? But Darwin had actually written about population size. And with Darwin, it's always about the pigeons. He's got these pigeons that have feathers on their feet and all kinds of weird things. And they like those pigeons in 19th century England. They compete at fairs. You can still find them at the state fair now, right? You've got these crazy pigeons. Well, Darwin says, how do you get the best animals? Well, there's one thing I know, says Darwin. You have to have a lot of them. If you're waiting for these weird things, you got to have a lot of animals, because these kinds of variations occur rarely. And Darwin didn't understand mutation. He didn't know about the mechanism of inheritance. But he understood that if you want to see rare things, you got to have a lot of them. And R.A. Fisher wrote about this in the 1930s. For Fisher, this was directly about a constraint on the process of evolution. Fisher was arguing with Sewall Wright. They both, together with Haldane, developed population genetics. But they had a fundamental disagreement. How important are random changes? Wright thought that random changes were very important. Fisher thought they were unimportant. Today, random changes are most of what we talk about. When you talk about molecular distances between species, almost all random changes. But Fisher wanted to focus on those things that were functional, that made a difference. And he said, as you get better and better adapted to your current environment, the possibility of adaptive mutations should get less and less, because the chance that you're going to get closer to the optimum from some change, it's less as you get closer to it. So for Fisher, this became a thing that would be complicated by population numbers. The more individuals you had, the better chance you would have to have an adaptive mutant. We sort of dug this up and developed some math to describe how the number of favorable mutations should respond to population growth in humans. And this is the pattern we're expected to see based on human population growth. Let me just explain what's going on. Here, more than 30,000 years ago, the population was small. So there were few favorable mutations for that reason. The other thing is that the ones that have happened, if they're very good, they've already spread through most of the population. As you go up toward the Neolithic, you're seeing a lot more of them until you get to some peak. And why does it fall here? It falls here because there's a limit to our ability to see stuff. We know that recently, every so often when we find them, there are mutations that have really cool effects. And we can't assess now whether they're fitness-bearing effects necessarily. But there are things like a mutation in Limone sul Garda, Italy, that made people relatively much less likely to get cardiovascular disease. And you go to this village and see all these old people buried in the cemetery. And they went to discover, and they said, well, there's this mutation. They're all descended from this person that migrated to the village in the late 1700s. That's an example of a really recent mutation. We don't know whether it made people have more kids. And that's the thing, is that when we get up here to more recent times, we'll never know. They're too recent for us to judge their evolutionary impact. And our power to see their impact declines as we go up toward the present. So what we've got here is a chart that predicts not what there should be, but what we should be able to see. It's an ascertainment chart. When we plotted this chart against the data, we said, let's find all the things that we can find. Well, let's make an estimate of their age and see how many of them fall into these age intervals. This is simulation data. Don't give me that kind of credit. We simulated this a bunch of times to see if we got the expected answer right. And this is what it ought to sort of look like. Yeah, wow. I thought wow too, but this is what the data actually look like. These are binned SNPs that we infer to be connected, linked to things under recent selection in two populations. The red is the Yoruba, West Africans, and the blue here is the CEU. These are Utah people, but inferred to be Northern Europe. And you can see that they both have that pattern that we expect to see, and that's encouraging. But the other thing is that there's a couple of demographic details that reflect what actually happened that sort of lend to the sense that this is actually telling us what's going on. One is that you can see there's a bunch more here in Africa. The African population was always bigger, until you get up to about 8,000 years ago, where you have a later peak in Europe. This is the early Neolithic population spike we would interpret in Europe. So it looks like this is what's going on. But a natural question, and furthermore, yeah, I don't want to explain that slide, because it's easier to explain this way. Furthermore, the thing is that if you count, there's a bunch of things now. If things were always happening at the same rate, if there were always 3,000 things proceeding to fixation in the human population, then if you compared us to chimpanzees, there should be many millions of things, 6 million things. There aren't. There are only maybe 12,000 things that separate us from chimps in amino acid substitutions. And if we count non-coding substitutions, regulatory mutations, the things that we are less able to see now, maybe there's double that number, maybe triple that number. It's not 6 million. So on that basis alone, we'd say, look, this is consistent with our explanation. The demographic forces have increased the number of mutations. This leads to a nice picture. If you just look at the number of years involved in the last 40,000 versus the human-chimp divergence, which is on the order of 5 million years ago, you can see that that's a tiny fraction of the time. It's a huge fraction of adaptive changes. If we just measured this time and said, how many adaptive changes is this? How many would we expect? We'd say that the number of adaptive changes that separate us from Australopithecus is probably around the number that have happened in the last 40,000 years. But that's huge. We can sort of categorize what's going on. And there's things that you expect, neuronal function, for instance. You say, well, okay, the brains are changing. Why are the brains changing? We saw that the brain size was changing. We don't know how that connects genetically, but evidently there's something going on with our brains. Protein metabolism, this is primarily dietary. Not lactase, that's sugar metabolism, but other kinds of dietary things. DNA metabolism, this may be related to aging. Reproduction, sperm. Sperm are crazy on this. I don't know what's going on with sperm. Let me give you some concrete examples. This is the distribution of the null allele at the Duffy blood group locus. Duffy is one of those blood types they don't test you for because it doesn't cause a problem with transfusions, but it does lead to rare incompatibilities. When they found it around the time of the Korean War, it led to rare, very rare incompatibilities in transfusions. So they characterized it. Today we understand that like ABO, there's a 3-allele system, and the null allele, the O equivalent, is very common, up to 95% here in West Sub-Saharan Africa. It's very rare other parts of the world. This is one of the big genetic differences between populations now. Duffy, we know, makes you almost immune to vivax malaria. And when we talk about how genes affect history, here's a population that has an allele we infer to be something like 30,000 years old. Vivax malaria, we infer to be around 35,000 years old. So it was a rapid and effective mutation. The places where Duffy is high frequency today don't have significant vivax problem. Vivax is the most common malaria in Thailand. You know, it's a malaria that's major in the Mediterranean basin. It's not here. It's not here because the people don't get it. Falciparum malaria has taken its place. You you can't, you can run as fast as you want to genetically, but you can't keep up with the pathogens. But what we're seeing is that a real effect on history. There are complexities involved in this, and I'm going to sort of talk about how hard this is, because when I talk about what the genes do, and the last talk sort of went through categorization, what the genes do, you have to understand that cases like Vivax, or cases like Duffy, are very rare, that we really, really know what the gene does. And if we have any experimental data on how it relates to disease, pathogens, diet, whatever. People are exploring this stuff now. This is a paper that came out last week, and I think it's interesting because these guys are looking for genes that explain blood measurable variables, like the number of platelets. You know, really gross, we measured this stuff. What they found is one region that has two SNPs that are very tightly linked across 800 kilobases, which means that they're selected. This haplotype that carries these is about 30% in Europe. I'll show you a map in a second. It looks like it's 2,000 years old, give or take. So it's a new haplotype. It's selected. It explains platelet count for some SNPs in this area. I'm showing you this slide to show you how complicated this is, because what we've got is an area. It's an area that stretches over some distance. There are SNPs in it that show up if you put them on patient case control studies. They show up as loaded on cases. So you're saying that these SNPs in this area are candidates as causative SNPs for platelet count, for coronary artery disease, for type 1 diabetes, which is autoimmune, for celiac, which is autoimmune. So you're saying that these are things that we think are connected. Why are all these things connected in the same area? Because they're linked. It's the linkage that makes them connected. We don't know which one causes it, if any, or if there's some combination in here that causes it. Furthermore, look at all the genes that are in this region. I'm going to show you the next slide that has a close-up on the genes. Here are all the genes. When I say that they more or less have to do with these categories, understand that in some cases you get something like this, where the selected haplotype in here could be anything having to do with alcohol metabolism. ALDH2 is a good candidate for this. There's BRCA1 associated protein over there. There's other things that are involved in cell metabolism. Who knows? Coming from the informatics end, saying we've found this area that has something interesting in it, and that area is loaded on disease-relevant traits, we still don't know what happened. But it helps somewhat to know where it happened. Here's this SNP that is tightly linked to the selected haplotype. Here's its geographic distribution. What's going on with this? The hypothesis presented in their paper, and I think it's probably one, is that there's something immune-related, some pathogen. If I had to throw out a guess as to which pathogen it would be, it's probably smallpox. But that's just a guess. How do you connect these things? I've been working on trying to connect these things by focusing on well-understood metabolic processes. I'll give you an example, hearing. We know of a lot of syndromic hearing loss disorders that there's a mutation that causes deafness. So we know the genes that are related to building the cells of the inner ear that receive sound, hair cells. That's convenient because this is a well-understood system. We know how they're connected to each other, and it's an old system. These have been receiving sounds since early vertebrate days. So they've functionally sort of worn in a groove. We know what these things are doing. Here's a hair on top of one of those hair cells. Yeah, I get no credit for any of these pictures, but they're cool. Here's another thing. A separate part of the hair cell system is that the hairs are embedded in this basilar membrane. There's another membrane that's on top of them called the tectorial membrane. And the way that they receive sound is that when the hair cells are touching this, it vibrates and they pick up an electrical signal. So that's the way that hearing happens. But there are mutations essential to building this membrane that if they rupture, this is a mouse ear, if they rupture, the membranes don't make contact and there's deafness for that mechanism. So we know what these genes are. Here's a list of recently selected genes that are in this category. things that involve hearing. And in particular, the hair cells of the inner ear in that system. So this is the reason why I focused on it, is that when we make a list of stuff that we think is selected, these things pop out as very highly loaded. This is a high proportion of genes involved in this system. We still don't know what they do. We've got some hints. I want to focus on PCDH15, partly because we know pretty well what it does. It makes these little links between the cilia, the fibers that make up the stereocilia. And we know a lot about its geographic distribution. Here's a gene that's strongly selected in East Asians in the last 10,000 years. Why? I don't know. We could throw out some hypotheses. Maybe this is about tone languages. That's a hypothesis. Maybe it's something that would have been adaptive 40,000 years ago but didn't happen. A lot of my things are like that. 40,000 years ago, it would have been better to have these stereocilia different, but it didn't happen because we're looking at rare adaptive changes. But we're catching up so that when the population grows to a critical level with Neolithic population sizes, you happen to get these good things once in a while, and they happen in some populations and not others, which is why we have these pigment variants that are different in different populations. It happened in Europe, didn't happen in Asia. The other one happened in Asia. This really comes back when I was starting in my PhD program. I was taught by Frank Livingstone, who was one of the people who really started to understand the malaria connection. What genes are adaptive in the case of malaria, the thalassemias, the sickle cell. And I asked him one day, Frank, it looks like hemoglobin E, which today is common in Thailand, looks like hemoglobin E is just better than sickle cell and C and all these things they have in sub-Saharan Africa. It doesn't kill people when you're homozygote. Why didn't the Africans get hemoglobin E? And he said, well, it didn't happen there. And that's really the message of how we're looking at this now. We've got hits. They happened because of chance. But they're all building into a picture of recent rapid demographic changes. And when we look at the lactase example, these are changes that contribute to the growth of populations. Lactase, the digestive allele, grew faster than the non-digestive allele. That didn't happen by the lactase drink, the milk drinkers killing off the other people. It happened because they had more kids. So the population grew faster than it would have without lactase. So the populations that had lactase grew faster than the populations who didn't. Okay, I'm going to stop there so that we have a few minutes for questions. Oh, just shout.
So in modern populations, where you have a gene which makes people live to be old, it doesn't necessarily increase selection. Aren't there sort of anti-selective forces?
So how does this deal with aging? Today we live a long time. Yeah, it doesn't make any difference at all. The stuff that makes you immune to cardiovascular disease when you're 70 doesn't make a bit of difference to fitness. The stuff that makes you more likely to reproduce when you're 16 might make a difference if it doesn't compromise reproduction later. There's stuff that, one of the things with cancers is that you get these mutations that sort of occur in pulses early in life, and then later they have manifestations. So maybe a cell cycle regulatory gene might improve your early life in ways that affect your later life. But yeah, this is highly loaded on stuff that, the things that we're studying with selection, highly loaded on stuff that make a difference to whether you have kids and make a difference to whether you survive pathogens and other mortality causes early in life. Yeah?
It seems that there may be some genetic variations that confer some resistance to HIV in sub-Saharan Africa.
Mm-hmm.
Isn't that an opportunity to study selection
Let me give you a quick answer, because my usual answer to this would be to Google and find out. But the quick answer is we know a lot about one that does it in Europe, the CCR5 delta 32 mutation, which is an HIV resistance mutation, but it's not there for that reason. It's there because in medieval times people were able to do it. So when we look at new stuff, we come down to this problem. How do we tell if it's making a difference? Today, in genomics, I think this is the real frontier of here's some new things, they were never selected before, but today they make a difference. How do you find them? Once in a while you're lucky, and it's something that happened to be selected before. Delta 32 is a good example of that. Happened to be selected before, today it makes a difference. But the whole principle of the HAP map, why have they gone from the HAP map to the 1,000 genomes? Because the HAP map failed. Its failure was more or less preordained. I mean, the idea was that by sampling a few hundred people, you were going to find common mutations that correlate with disease. I've got to tell you, any evolutionary biologist could tell you that common mutations that correlate with disease should not happen. How do they get to be common? If they kill you. Now, once in a while, they do something good at the same time, right? So once in a while, like sickle cell, you get something that gets up to some substantial frequency and it happens to have a good effect. And we find some of those. It's a great credit to humanity, I guess, that we've evolved so fast and had long linkage blocks that happen to correlate with things. But it shouldn't have worked. So the 1,000 genomes has the promise of finding these new rare variants that aren't widely shared, but might have happened multiple times. But it's going to take a lot more than 1,000 people to find this. It's going to take tens of thousands, maybe hundreds of thousands of people to find these things that load on diseases because of shared mutations that are recent.
I had a question.
Yeah.
You correlated shrinkage in skull size with shrinkage in brain size. Couldn't the brain just be folding more? I mean,
oh, so yeah, I didn't go into the detail that I might if I was teaching undergraduates, right? But well, because they always give the best answers. I asked them, why is the brain shrinking? And of course, the number one answer, we don't need it anymore. What do we need these brains for? Yeah, we've got iPods. So yeah, we don't know. Yeah, the fundamental principle is that maybe, I'll give you the hopeful explanation. The explanation I think, on my good days, I say, here's what's going on with brain shrinking. What's going on is we're not on our way back to Homo erectus times. We're actually getting more efficient. Big brains are bad because they're energetic. This is like a 20-watt light bulb on your head all the time. And in the context of your energy budget, that's a lot. So if you're living in a famine-prone population, as all early Neolithic populations were, the best thing to do is to have small brains. Besides which, small brains develop faster. So if you want to reproduce when you're 16, 17, 18 years old, the best way to do it is not still be growing your brain when you're 16, 17 years old. So for all those reasons, say that efficiency demands that the brain should be smaller, and if you could find a way to do it in a smaller space, that would be highly selective. So maybe we've gotten better with smaller brains. But I got to tell you that maybe we're getting dumber. I mean, how can we know, right? I say on a good day, I think we're getting more efficient. On a bad day, I say, it's idiocracy. We're on our way.
On that very positive note, thank you. Dr. Hawks is going to be around on campus over the next couple of days. He'll be at lunch. So if people